조절바 타나자의 초나디피 바하 바주 메주 빈더 토니코구 쿠시주야 청구 구티 seminar Newahs in British Contemporary Society Sub Lasa Kusa Yana Tona. Ladies and gentlemen, good morning and welcome to Guti Seminar 2015 under the theme of Newahs in British Contemporary Society. My name is Triveni Gurum. I am the General Secretary of PPGUK UK, the Vice Coordinator of today's event, and the convener for today's seminar. Firstly, I would like to start off with some housekeeping. Please, if you could have your mobile phones switched off or on silent. The toilets are located through the barriers onto your left as you exit out of this hall. In case of an emergency or fire alarm, the emergency exits are just located on the sides here. I would now like to invite PPGUK UK's President, Mr. Speech Shrestha, who would like to share a few words.
타나자이디피 포감 바주 마이주피 जुजलबा आजको यस सेमिनारमा उपस्थित सम्पूर्ण महानुभावहरूलाई PPGUK UK लन्डनको तर्फबाट म हार्दिक स्वागत गर्न चाहन्छु। आजको यस सेमिनारमा विभिन्न विधाका विज्ञहरूबाट प्रस्तुत हुने मन्तव्यबाट हामी सबै लाभान्वित हुन्छौँ भन्ने कुरामा विश्वास लिएको छु र पुनः एकपटक तपाईहरु सबैलाई स्वागत गर्दै बिदा चाहन्छु। शुभकामना। धन्यवाद।
I would now like to explain the structure of our presentations for today. We have 10 speakers who will be presenting today. They will be presenting either their academic background or their professions. There will be 3 sessions. Each session will be chaired by the session chair. Just to let you know, we've already nominated the session chairs. Presentations will last for 20 minutes with 5 minutes of question and answer session from the audience. The session chair will signal when presenters have 5 minutes left. We would highly encourage you to interact with our presenters during the lunch and tea breaks. In order for you to identify the presenters, we have their introduction slides or their ID slides just placed over here on the walls. Okay, so let's begin our session 1. In this session, the following speakers who will be presenting are Dr. Nikki Sakya, a Speciality Registrar in General Adult Psychiatry at Rotherham and Doncaster NHS Trust. Speech Shrestha, a biomedical scientist currently working in the Blood Science Department of Papworth Hospital, Cambridge. She will be presenting the role of a biomedical scientist and how they are significant in analysing biological specimens, which help clinicians to reach the correct diagnosis. Sudhin Kansakar, a consultant at GE Healthcare. He will be presenting his experience of teaching and consultancy and the things he learned throughout his career and how his careers are interlinked. And lastly, for this session, we have Sonu Shrestha, who is a research assistant currently working with Oxford Vaccine Group at the University of Oxford. Her presentation will explore the issue of human diseases, vaccines, and also some areas of clinical trials. I would now like to invite Mr. Sashidhar Manandar, one of the founding members of PPGUK UK, to chair this first session.
Before I request Ms. Sonu Shrestha, I just want to say one sentence, and that is, PPGUK, Passaputo Guti, formed in 2000, and the first seminar we conducted in 2003 related to Newahs culture, language, and society. And I'm very pleased to know, after the first seminar we organized, the younger generation are continuing with this process and organizing seminars highlighting the Newahs young people and their studies in science and art. So thank you for coming and coming to the seminar. So can I invite Sonu Shrestha on infectious diseases and vaccines to give a presentation. Thank you.
Namaste, my name is Sonu Shrestha and I'm going to present you with the infectious diseases and vaccine today. I would firstly like to thank PPGUK on inviting me to present this slide on this particular seminar day. So basically, I am a microbiologist and I was working in a molecular diagnostic center in Nepal, which mainly worked in a PCR and diagnosis of HIV and other tropical diseases in Nepal. So currently I'm working as a research assistant in Oxford Vaccine Group. This particular Oxford Vaccine Group is a place where different kinds of clinical studies occur, and we mainly focus on pediatric and adult vaccine studies. Currently, we have a following active clinical studies going on, which includes understanding the vaccines' efficacy on typhoid and paratyphoids, effect of different vaccine regimen studies in children, as well as RSV, which is respiratory syncytial virus study, as well as efficacy of current Johnson & Johnson's vaccine clinical trial in our organization. Before moving into what infectious disease and vaccine is, the basis of infectious disease lies in a tiny organism called as microorganism. It is a tiny living entities that generally cannot be seen with our naked eye. So they are mainly categorized or broadly categorized into 4 different groups: virus, fungi, bacteria, and protists. In this particular definition, I'm question marking these 2 sentences, like living entities and generally cannot be seen with our naked eye, just because living entities, in a sense, because all organisms like bacteria, fungi, and protists follow the rule of living organism, like breathing, they can eat, they can propagate. But in this case, the viruses falls into that particular entities which is neither living nor non-living. So, so a particular virus, they need a particular cell to grow, like our cell to grow and propagate. That is why it is called as entities, and it's still a controversial area. There are certain organisms like Margarita nambibiensis, which is also called a sulfur pearl of Namibia, and which is still a single cell but can be seen with our naked eye. That is why I tagged generally cannot be seen. Not all organisms cannot be seen, but this particular organism can be seen even though they are a single cell. So where are they found? As we know, microorganisms are found everywhere: air, water, soil, food, everywhere. Even when you breathe, you're taking in, and when you're expelling out microorganisms as well. Not only that, it is also found in our most extreme areas like in Antarctic, where there is the propagation of life is hard to think about, in desert as well as in Antarctic lake. Not only that, some of the scientists have discovered some microorganism in the spacecraft, which is considered to be a clean room as well. So it is present everywhere, it is eminent. Not only that, it is present inside us and on us as well. So, yeah. So they can be broadly categorized into two groups, like good bacteria and bad bacteria. Good microbes, they help in producing different kinds of foods like bread, probiotics, that helps in evading or that helps in killing microorganisms, bad microorganisms away, while bad microorganisms, they help make you sick or make you ill. So these bad microorganisms are the one that causes infectious diseases. And these are the list of microorganisms, some of the list. It's not the only list, but it is just considered as a small list of each category of microorganism and what they cause and diseases. So this particular slide, the chain of reinfection, is the one that describes about how the microorganism gets transmitted from one to another. When a microorganism enters one's body, like for example your body, you act as a host to it. So it starts growing in your body, and then when you sneeze or when you cough, and when you start transmitting it to the others, so it exits from your body and enters into a susceptible host, and this particular cycle keeps on happening, and this particular chain of reaction keeps on happening until the microorganism, the idea of microorganism living is to continue this chain of infection. So more for disease transmission, the microorganism can be transmitted through contaminated food, like typhoid. It could also be transmitted through vectors like mosquitoes. For example, a disease called malaria is through mosquito transmission, then HIV are sexually transmitted diseases. Infections are either symptomatic or unsymptomatic and could cause fever, mild to acute diseases, chronic, or even can to get death. In a low-income setting like our country, it is hard hit, while in a developed country like UK, it does still exist in here as well. So this is a very funny picture about how our body responds to a microorganism. So as you can see, when a bacteria, not using this pointer. So when an antigen or a bacteria, virus, or protozoa enters our body, it has to go through our first line of defense, called as the skin or tears, and this particular acts as a barrier towards microorganisms. So when a microorganism tries to enter, our skin protects us. Similarly, our tears. So if something goes into our eyes, our tears come out, and those tears consist of an enzyme called lysozyme, which kills the microorganism. If you ingest something, like for example contaminated food, the microorganism can be killed in your acidic condition in your stomach. And when you breathe in some organism, you can have certain reflexes like sneezing or coughing. It helps you expel your microorganism out. So this is the first line of defense that our body tries to eliminate those organisms away. If the organism passes through this particular first line of defense, either through abrasion or it still goes inside your lungs, it comes into contact with various kind of cells, like natural killer cells or phagocytic cells that tries to guzzle it up, eat it up, and then break into pieces so that it can be taken out from our body. In these processes, you can get inflammation and fever, which increases the temperature of our body, and some microorganisms cannot tolerate that high temperature, and thus can be killed. The microorganism, when it passes through this particular second line of defense as well, it comes across to the third line of defense, where kind of FBI-style T cells and B cells are present, which helps in taking those microorganisms out, killing them, or eliminating them from our body. In this process, antibodies are formed, which stays in our body as a memory. So vaccine also works in this way, where the efficacy of vaccine, or whether the vaccine is working or not, is evaluated by testing whether T cell and B cell is producing these antibodies or not. So the infectious diseases are treated by antibiotics, antifungal, antiviral, antiparasitic drugs, as well as some food supplements like probiotic. So first and foremost, prevention for any kind of infectious diseases is adopting a hygienic, safe, lifestyle, and eating safe food and clean food. And most importantly, the most advanced way of preventing yourself from any microorganism is through vaccines. So what is vaccines? Vaccine is a biological agent that resembles the part of a microorganism. This microorganism is not alive. In a vaccine, there is a part of a microorganism or a whole of a microorganism, but this particular microorganism is not alive. They are either killed or their certain part is taken and then injected. So when this particular vaccine is given to a body, it mimics a kind of infection. So the body identifies the foreign body, the microorganism as a foreign body, attacks it, and attempts to kill and eliminate it out from our body. It pushes it out. In the meantime, it produces the antibody that I explained it to you before in slide. It stays as a memory in our body. So when the second time the real microorganism comes and attacks your body, it efficiently and very quickly tries to eliminate from your body. So there are various kinds of vaccines. There are a lot of vaccines, and since I work in a typhoid vaccine, these two vaccines are very commonly used. And typhoid VI is a part of a VI antigen. This is a salmonella, and which causes a typhoid in human being. And VI antigen is a virulence factor, which is taken, and then this particular vaccine is produced, which is injected in our body, and it produces a similar kind of reaction, and then and even the memory is created. So another vaccine is TY21A vaccine, or oral vaccine. These are consumed orally, and these are the whole microorganism, but the main virulence factors is taken out. And that is why it is safe, but it is more effective than these ones. There are numerous other vaccines as well beside these. So when a vaccine, how does a vaccine from a lab reaches to a rack of a pharmacy? There is a whole process of, long process of creating a vaccine and then it reaching to the pharmacy. It takes somewhere around, if you add up these years, it takes somewhere around 15 to 18 years from start to the end. So one particular vaccine to go from a lab to a pharmacy takes around 15 to 18 years. So firstly, it starts with identifying those virulence factors like VI or microorganism and producing them, then preclinical testing them in mice or testing them in cell culture. And then the human volunteers starts from phase 1 and it proceeds to phase 3. The efficacy and safety is important part during phase 1, phase 2 and phase 3 vaccine trials. Currently we are in our organization where I work, some of our group is working on finding these antigen for other organisms like meningitis, while in typhoid